We're going to pull together a lot of big ideas this week. We're going to use our understanding of DNA and transcription and translation, our knowledge of how a change in a DNA sequence could affect a protein in an organism. We're going to combine that with our understanding of meiosis, and particularly how homologous chromosomes separate during meiosis, to build a deep understanding of genetics this week. And in doing that, we're going to be concerned with two key geneticists, Mendel and Morgan. So we'll start with Mendel. He's first and is often called the father of genetics for all that he's taught us. Mendel's working in the 1800s on pea plants. He likes pea plants because he's got true breeding varieties available that have different traits he can observe easily. In his most famous experiment, he crosses a true breeding purple flowered plant with a true breeding white flowered plant. He calls these his P1 generations, or the parental generations, and notices that all of their offspring are purple. Mendel's hypothesizing that there are discrete units of inheritance. This is a new idea for the scientific community, that discrete units of inheritance get passed down from parents. So he thinks you get two of these, one from each parent. Sounds like homologous chromosomes, doesn't it? One from each parent. And if you got two different ones, in the case of those offspring between the purple flower and the white flowered plant, one of them is going to control what you look like. He calls this the dominant allele. So in pea flowers, we could say the purple allele is dominant to the white allele. If you have one of each, you'll be a purple flowered plant. So he takes these offspring of his two parents. These are called the F1 hybrids. They're a hybrid because they have a mixture of these two genes. And he crosses them to each other to give him what he's calling the F2 generation. And in this F2 generation, he observes something very interesting. A really strong ratio of 3 purple flowered plants for every one white plant in his offspring. And he's looking at hundreds and hundreds of offsprings and seeing a really predictable outcome. So to Mendel, that says there is really something discrete and predictable about genetics and inheritance. So we want to represent that, because we're going to think about a lot of traits this week that are inherited exactly as Mendel would have predicted. And we've got a powerful tool to help us understand and predict that. It's called a Punnett square, and it looks something like this. So what I've represented here with my letters are the genotypes, or the combinations of genes, alleles, at a particular spot in an organism. So I'm representing it with a capital P to indicate this is the dominant allele. In the case of flower color, this is the purple allele. And this plant here has big P, or 2 purple alleles. I could also call it homozygous because it's got 2 of the same alleles. I'm going to cross it to a plant whose genotype I'll indicate as little p. This homozygous plant has 2 recessive alleles. It's going to have white flowers. So these are Mendel's P1 generation, the true breeding purple flower. He's going to cross with the true breeding white flower. I'm going to represent this now on a Punnett square. And to do that, I'm going to think about meiosis and think, if this is a female plant, for example, which one of these chromosomes could she pass on to her eggs during meiosis? And I know that her eggs are either going to get this chromosome with its dominant allele, shown right here, or this chromosome with its dominant allele. So I'm going to show all the females' possible gametes or sex cells here. Then I'm going to think about my male parent. He is homozygous for the recessive white allele. So I know that all of those pollen grains are carrying either this recessive allele or this recessive allele. So I'm going to show all the possibilities across here. And then what a Punnett square lets me do is fill in the possible combinations. So I could imagine it was this pollen grain fertilizing this egg, and think about the genotype of that offspring. It's going to get the dominant allele from mom and the recessive allele from dad. This flower now is a heterozygote. It's got 1 of each allele. I could keep filling in my chart then and say, well, what if it was this sperm that fertilized this egg Here we go. You've got another heterozygote. Likewise, I'm going to fill in what if it was these two that met up, or what if it was these two that met up. So what I've got represented for myself here is a Punnett square that shows me the F1 generation. My parents and the F1 generation in this cross. All of the F1 generation are heterozygous. They're all purple flowered plants, exactly as Mendel predicted they would be. So we've got a nice tool here for visualizing this process. Mendel takes this one step further, and let me show you what that Punnett square would look like. He's now going to create an F2 generation by crossing two of these plants with each other. So to get this F2 generation, I'm going to do something very similar with my Punnett square. I'm going to take two of these heterozygotes and imagine them as my parents. So I'm going to think about the female here, knowing that her eggs are either going to get this dominant allele or this recessive allele, and I'll show both of these possibilities in my Punnett square. Likewise, for Dad, it's going to get either the dominant allele or the recessive, and I'll show both of these possibilities in my Punnett square as well. So this time, I can again go through and fill in all possible offspring. What happens if these two meet up and undergo a fertilization event? What happens if it's these two that meet up and undergo a fertilization event? Likewise, and here we go. So I'm filling in the genotypes for all possible offspring from this cross. And what I should notice is that three of these give me a purple colored flower, right? One of them, only that plant that's homozygous recessive, will have a white flower. So the Punnett square very nicely explains that powerful 3-to-1 ratio that Mendel observed in his offspring. It also allows us to point out a critical difference between genotype and phenotype. So here I'm showing you genotypes when I describe to you what that organism looks like, which is really a combination of its genotype and environment. I'm talking about phenotype. So our genotype here might be homozygous dominant or heterozygous, but phenotypically both of these are purple colored flowers. So that's one powerful lesson we get from Mendel's work. Inheritance is predictable. I can use that tool of a Punnett square to show me what I expect to see in the offspring of any cross. Mendel takes this one step further to do a dihybrid cross. He wants to look at two genes and see if they're inherited together. So for this experiment, he follows a gene for pea shape, and peas can be round or quite wrinkled, as well as a gene for pea color. They tend to be yellow or green. He looks at these two alleles together, does some interesting crosses, and decides that individual genes are inherited independently of one another.